Hi, my name is Brian Glowski, and this summer I worked on a research project titled The Development of a Transformation-Free Method to Produce Therapeutic DNA Minicircles, which is centered on the production of DNA minicircles for use in gene therapy. Gene therapy is a technique to replace, repair, or regulate genes to treat a disease. This project focuses on the production of plasmids, which are small, generally circular, double-stranded pieces of DNA as vectors for gene delivery. Plasmids are ideal because they carry a small number of genes and are easily manipulated to remove or add genes of interest. The genes mCherry, which is a bright red fluorescent protein, and Gaussian luciferase, which is a bioluminescent luciferase enzyme that oxidizes substrates to emit a blue light, were used in the production of a parental plasmid due to their ability to visualize protein expression. Seamless ligation cloning extract, abbreviated SLICE, is a novel cloning method that utilizes short end homologies between vector and insert and was used to clone the mCherry gene into a vector backbone, producing the parental plasmid. Then, the minimum expression cassette, which is the smallest sequence of DNA needed to express a gene consisting of a promoter and a gene of interest, is PCR amplified from the parental plasmid as the first step in producing our minicircle product. Minicircles are the focus as vectors to deliver genes in gene therapy because of their ability to avoid immune detection, inability to confer antibiotic resistance to human microbiomes, and they show increased shearing resistance along with improved transgene expression. We began this project with the production of a parental plasmid containing the mCherry gene. To start, we PCR amplified the mCherry gene and a vector backbone from two separate plasmids, introducing short nucleotide end homologies between vector and insert by primer overhangs. Next, we purified the PCR product through gel purification and quantified the DNA concentration of the purified PCR fragments to prepare for the SLICE reaction. SLICE was used to clone the mCherry gene into the vector backbone using the short end homologies that were introduced by primer overhangs in PCR. SLICE seamlessly combined the vector and insert into one circularized plasmid product named P-mini parental mCherry, shown here. This SLICE product was then transformed into chemically competent E. coli cells and grown on an LB agar and ampicillin-laced plate to select for the parental plasmid containing bacteria. Following this, 3 colonies were chosen from the SLICE product plate and colony PCR was performed on them. This involved amplifying the mCherry fragment from the plasmid inside the bacteria to select for the colonies that contain the mCherry gene. All 3 tested colonies came back positive for containing the mCherry gene. Lastly, to validate the overall size of the recombinant plasmid, a restriction enzyme, BBS1, was used to digest the parental plasmid into 2 linear pieces. The linear product sizes were as expected, as the sum of those were equal to our parental plasmid size of 4,302 base pairs. After production of our parental plasmid containing the mCherry gene, our next step was to optimize the PCR amplification of the minimum expression cassette from the parental plasmid containing the Gaussian luciferase gene. Initial amplification resulted in a low-intensity DNA band, which indicated a low PCR reaction efficiency. To optimize this, we increased the molar ratio of primers. This involved a 2-to-1 ratio of forward to reverse primer, respectively. Since our primers used in PCR are complementary to each other, this promoted the formation of primer dimers, which is when the 2 primers bind to each other instead of the DNA template, which ultimately lowers PCR reaction efficiency. By doubling the concentration of the forward primer, this allowed for more primers to be in solution to bind to the DNA template, and PCR amplification could continue. The result of this 2-to-1 ratio was a significantly stronger PCR reaction efficiency and a much brighter DNA band intensity. In conclusion, the mCherry gene was cloned into a vector backbone, resulting in the production of a parental plasmid called P-mini parental mCherry. The parental plasmid was selected for through the process of transformation in colony PCR, and its final size validated by restriction enzyme digestion. In addition, the minimum expression cassette, termed P-mini GLUC, was PCR amplified, and the PCR reaction was optimized. Moving forward, the P-mini mCherry minicircles need to be produced and purified to transfect mammalian cells for visualization of fluorescent mCherry protein production. I want to give a special thanks to my colleagues in the Harada lab for their patience and guidance on this project and constant support. Additionally, thank you to the Institute for Quantitative Health Sciences for the undergraduate learning opportunity and the Ensure program for providing financial support on this project.